Hello and welcome to the Genetics Podcast. I'm your host, Patrick Short. My background is in population genomics and studying the genetic causes of rare disease. I did my PhD at the Sanger Institute in the University of Cambridge and have been in biotech since 2018 when I started Genetics. Genetics helps academic and industry researchers to run large-scale genetic testing programs that speed up their clinical trials, generate datasets for the next big breakthrough, and give participants the best possible experience taking part in research. Each episode of the Genetics Podcast, we bring you insights from the leading minds in genetics and precision medicine, including household names and Nobel Prize winners, as well as early career scientists and biotechs working on the next big breakthrough. Whether you are a scientist, entrepreneur, executive, patient advocate, or simply someone curious about how genetics shapes our world, you're in the right place. Thank you for listening, and let's get started. Welcome back, everyone, to the Genetics Podcast. Today, we're joined by Jonathan Marchini, who is a pioneer in statistical genomics, and he is today the head of statistical genetics and machine learning at the Regeneron Genetics Center. Before he joined Regeneron, he spent 2 decades as a professor at University of Oxford, where he worked on really foundational tools that have become standard across the industry and have driven many of the main projects that we still look to today as landmarks in the field. Wellcome Trust Case Control Consortium, 1,000 Genomes Project, and of course the UK Biobank, and many more. So Jonathan's work has enabled discoveries at a global scale and helped bring us from the early GWAS era into the million-plus exome era that we're in today. So Jonathan, I've been wanting to do this episode for a really long time. Thank you. I'm looking forward to the conversation.
Well, thank you very much, Patrick. It's a pleasure to be here. I'm happy to chat to you.
I want to start with your origin, actually. Can you share how teaching math in rural Tanzania, which is, I think, one of the first or maybe the first job that you had, led you to genomics? It's an interesting and winding path that I think people would love to hear more about.
Yeah, it's a bit of an unusual story. I guess I did a maths and stats degree at university, and I guess during that time started to think about what I wanted to do with my life, my career. And, I guess I got interested in the idea of research. I also really liked the idea of teaching, and the idea of being an academic really appealed to me. And so that was kind of a long-term plan. But the idea of going out there, exploring the world, teaching also appealed to me. And so I found a route to do that. I became a VSO volunteer and taught maths at A-level rural school in Tanzania for 3 years, which was a life-changing experience for me. I think I gained a lot more than I ended up giving, but and some amazing friends. And then after that, I kind of pivoted back to this idea of wanting to be an academic, a researcher. So, of course, did a PhD. I was very fortunate to get a place at Oxford, working with Brian Ripley on brain imaging. And so did that for 3 years, learned an awful lot. And then I guess the natural thing for me to do would have been to stay in that field, or statistics more generally. But I had this real sense at the time that genomics was really at the kind of inflection point. The technology and the Human Genome Project had just been published. It was a really exciting time. And so working with big brain imaging data sets gave me a lot of experience that I thought could be useful for analyzing big genetic data sets. And so talking to Peter Donnelly, Lon Carden, who were there at Oxford at the time, advised me to apply for a Wellcome Trust Fellowship, training fellowship, which I got. And then that kind of went from there. And then kind of in the right place at the right time, working with people like that, really fortunate to be involved in projects like the HapMap, International HapMap project. And then that kind of led into things like Wellcome Trust Case Control Consortium, 1,000 Genomes, and a really kind of talented group of people in Oxford and also around the world. I think one of the things I think back to that time is how projects like the HapMap really brought the field together, right? There'd been this realization that the kind of standard paradigm of family-based studies really wasn't working. Replication rates were low, and these realization that we needed to kind of pivot to association studies. I guess that's...
Yeah. If you take us back to that era, the HapMap, Wellcome Trust Case Control Consortium, 1,000 eras time period, what were some of the greatest surprises or most interesting stories in your mind that come from that period of time? Because you learned a lot in a rapid period of time, right?
Yeah, well, like I said, the Human Genome Project had been laid the foundation for this by effectively understanding a small number of people's genomes. But what we didn't really know was how that varied across people and groups around the world. And the technology had advanced, and so we had the ability to go deeper. And the papers like Regeneron back in 1996 really kind of said, look, if you could do something like 1 million SNPs across the genome and assay them for association directly with a phenotype, that would have much more power. You need much lower sample sizes than if you did a family-based study. I think they compared it to an effective SID pair design in that paper. And that shifted thinking quite a lot, but we didn't really know how to do that.
Yeah.
We had this idea of 1 million SNPs was just kind of not fully developed. We really needed to understand how many SNPs there were, how correlated they were with each other, how that varied across populations. And we had some idea of how recombination varied across the genome from family-based studies. There was some evidence, small scale studies that showed that there was maybe more LD than predicted by simple population genetics models. And so that formed the basis of this HapMap project, where they tried to 270 people across 3 continents, about 1 million SNPs, I think, in the first instance. We really learned in detail across the genome how LD was structured. Had this block-like LD pattern. It's prevalent. And then colleagues like Simon Myers, Gil McVean, Peter Donnelly worked out how to estimate fine-scale recombination rates from that data. And that's really the driving factor behind this LD block structure, recombination hotspots, etc. And I think that was a major advance there. And then this data formed the basis for people being able to design in detail US chips, genotyping chips.
Yeah.
And companies like Affymetrix and Illumina iterated on that. And then along us and other people like Gonzalo Abacasis and my colleague at Regeneron were thinking about how to use this type of reference panel data, came up with the idea of imputation. So I think not many people maybe saw that coming.
Yeah.
And of course, that's been very influential and successful, this idea of being able to predict the genotype completely.
That's interesting. So that wasn't in the water and came as a little bit of surprise, the idea of imputation.
Well, it kind of developed gradually. I remember in the HapMap project, we were trying to think about, given a set of SNP data in a given region, could we try to understand the distribution of SNPs that hadn't been observed? Yeah, this program, I remember, called the Hidden SNP Simulator, working with Peter, Gill, and Simon on this. And I guess in discussions about that, I think we realized that, well, if you had a reference panel of SNP data, well, and it didn't have some of those SNPs in subsequent data, you could use similar models there in that setting to predict or impute unobserved genotypes. And so, but there were lots of people, like I said, Gonzalo and others, thinking along similar lines. It kind of came together gradually, I think.
Yeah. Are there big challenges that you all had at the time that actually are still with you today? I'm especially interested in ones where you're like, it's been bothering you for 10, 15, 20 years, if we can go there.
I'm not sure I have anything massive that stands out. I mean, when I think back, what happened there was lots of people came together to work on this, which wasn't happening, I think, at the time. And that's really, persisted. People now collaborate a lot more than they used to do for meta-analysis in GWAS studies. Data sharing, I think, has improved a lot. I mean, it's not perfect. Building tools that people can use easily, I think, is still very important. Yeah. And I'm not sure it's fully solved. I mean, I won't mention the method, which we've been trying this as a Regeneron. We've been trying to get it running. It's really difficult. We've kind of given up because the software the paper that describes what it does is very nice. The software is very hard to use. I think that's something I've focused on, I think is very important. I spend all this time working on a method. Yeah. And it's really nice if people use it, and you have to make it easy for people to use. And you have to think about that from a user perspective.
Do you think we still underinvest in software engineering and like productization of these kind of things, for lack of a better term?
Yeah. I mean, I think good low-level coding skills are really important, and maybe some more development there, some more investment there could be useful. People like to do things quickly and easily.
Yeah.
You've got to facilitate them doing that, and writing good, clear code really helps. I don't code so much anymore, but, got amazing colleagues that do that really well. And it's just, it allows you to iterate so fast, right? And make advances if you can do that.
Bringing us maybe into the world where we are today, it's really different in the sense that you've analyzed more than 1 million exomes at Regeneron Genetics Center now. There's a landmark paper that came out last year that I'd love to talk a little bit about. When you think about that work, I almost want to know, like, where do you start? Because you've done brain imaging, you have phenotypes, you have genotypes, you have proteomics, you have all kinds of different sequencing assays. When you're thinking about how you wrap your arms around these large data sets, how you make sense of them, and how you do something that can help you accelerate therapeutic development, where do you start there? And maybe you can talk about some of those technical hurdles in particular that you face.
Well, I can describe what happened when I joined Regeneron and how things developed there. What Regeneron have built with the RGC, I think, is pretty amazing. I was really attracted to go and work with the team there. And the ability to create such large exome sequencing studies together with really deep phenotyping data is crucial for making drug discoveries. But the scale of the data was and is, I think, unparalleled. And that comes with really big computational hurdles. At the time, we were running existing tools, but we could see very quickly that they were going to hit a bottleneck. And so in developing this method, Regeni, that we still support, really was the idea that you want to run these linear mixed model approaches to do association studies, but they don't scale particularly well. And we're just trying to think about how you could fit those models quickly. We were thinking about, how you could fit these models in a low memory way. And, at the core of them, and the core of these linear mixed models, which are very similar to whole genome regression models, there's this idea that first step is estimate something like a polygenic risk score, which you then use to condition on when you do the actual tests. And so how can you generate one of those quickly? And so having a background in thinking about how SNPs are correlated across the genome, this idea of splitting the gene up into small chunks, estimating a mini polygenic risk score, and then kind of adding those up across the genome has the advantage of being very quick to calculate, very low memory, and it ended up working quite well. I don't think I knew if that was going to work, but it was a good idea to try, and had the pleasure of working with my colleague Joel Mbachu, who's the lead developer of Regeni, who really kind of took that idea and has run with it. People like Andrei Ziadinov has done some really exciting work there to extend it to some of the burden test work we've been doing. And it's allowed us to, one thing about that model is a lot of the computation can be reused across phenotypes. So then this idea that you can do more than one phenotype at a time. And then thinking about how, when you do burden tests in exome-wide studies, you really rely on annotations. You've got to make a decision about which variants you kind of roll up into a test. You really want flexibility there. And so you're then building out the code to allow different tests, different annotations. I think that's been quite successful and useful to people. Definitely has been for us. And annotations is missense variants in exome studies is a very active field, and new annotations coming out all of the time. You want to be able to pivot quickly and save compute when you can, because we're analyzing millions of samples, tens of thousands of phenotypes. Redoing everything when one little thing changes is potential for a real bottleneck. So part of my team spent quite a lot of time thinking about that. And then it's led on to trying to do meta-analysis quickly with this new package we have called Remeta. And I guess the insight there is that you can do these burden tests from summary stats. It's a single variant summary stats and effect size if you know the LD between them. And then can you do that quickly? So we worked out that if you have a reference set of LD from a cohort, we can take that LD matrix and just adjust it effectively to any kind of subset of individuals that you've run your XWAS on. And now we're using that as part of our engine. So you develop the methods when you, I guess throughout my career, been doing things for the first time. HapMap 1,000 genomes, Wellcome Trust Case Control Consortium, Biobank, now at Regeneron. The data sets have been growing, the problems arise, and you have to think hard about how to solve them.
Yeah, and you create these new methods, and it's hard to predict actually once they get applied, what interesting is going to come out of it, right? Like if we take the million-exome paper, what was the most interesting thing from that paper for you once some of these methods started to be applied at that kind of scale?
Well, the million-exome paper really showcases the breadth of the data sets that have been collected at the RGC. We collaborate quite broadly with healthcare systems, academic groups, biobanks to sequence at scale. And you got to the point of having reached the 1 million mark, and we thought this would be a really interesting genetic variation survey paper. And so 1.1 million PLOFs were discovered across that data set, varying ancestries. 50% of those are singletons, which I think is something that is very important. You can't get singletons by imputation.
Right. Yeah.
So they're not.
Just sorry for the listeners, those are predicted loss of functions, right? PLOF.
Yeah, PLOF, sorry, predicted loss of functions. Yeah. And so rolling those up into burden tests is extremely useful. Imputation can get you so far. I mean, with a reference panel of about 500,000, you can probably go down to around allele count of about 10. Appreciable imputation accuracy, maybe a bit further now. So just that understanding about 10 million missense variants in that data set across populations. There were around 5,000 genes with at least one human knockout. Really interesting. And then about 4,000 genes that were clearly very depleted of PLOFs. So those were interesting discoveries. And of course, now we have nearly 3 million exomes.
Really?
So,
wow.
Yeah, I think it's around that. I don't know the precise number.
What has driven that scale up? Is it just more and more collaborations?
More and more collaborations, yeah. And it's ongoing. I mean, I think the lab has the capacity per year to do 100,000 to 1 million samples.
Wow.
I don't think we hit that every year, but we're working hard to collaborate with people, and the model is work well. Effectively, what we bring to the table is the exome sequencing at scale. What we try to collaborate with is the phenotypic data, the samples and the phenotypic data, and that's been very attractive to people. I mean, it's on a well-trodden path now, and all credit to colleagues at the RGC and Regeneron for thinking about that and putting it in place and investing.
Yes, and the methods you've developed, where do you predict they'll start to hit a ceiling computationally or otherwise, and you need to go back to the whiteboard?
We operate via meta-analysis effectively. So it's the combination of biobank is huge, of course, but most of our data sets aren't quite that big.
Yeah.
And we can analyze them quite reasonably. And so then as I said before, with Remeta, it's this idea that how to speed up that meta-analysis step without having to go back and redo things when annotations change. Yeah. So I feel like we're in good shape, but we constantly trying to change things. I mean, at some level, it's about reading in the data as quickly as possible.
Right.
And then it's about the statistical methods and how, if you've got quite a complex statistical method, you can fit that quickly and efficiently. And so it's the combination of the two. So we're constantly trying to think about file formats and those methods to do that. I mean, GPUs are something that constantly comes up. What about moving things to a GPU? But, they are faster, but they are more expensive. And at the end of the day, it's our compute cost that we need to control. And so it's not like this switch to GPUs and it all becomes a lot better. But we'll see. I constantly think we've hit the limit on what we can do. I'm talking with Olivier Delano about kind of history of imputation and phasing. I think all through our career, we thought, we've hit the best we can do. And then this new idea, us or someone else, that moves the field forward. So take it year by year.
Yeah. While we're on the topic of imputation, one of the first things I think about is polygenic risk scores, and you and your colleagues at RGC have done a ton of interesting work around this. I want to ask you about polygenic risk scores in general. Where do you see them having the most impact in the near term based on the understanding we have today? And if you want to talk about as well, medium or long term, what might need to happen for them to be even more impactful? I'd just love to have your perspective on that.
So yeah, I mean, I think this is a really exciting field. I mean, I think maybe people are trying to translate these into the clinic. I think that's ongoing, and I guess going to be quite a slow process for them to be adopted. I mean, I think they kind of really do clearly stratify people according to risk. It's then, can you actually translate that into saving lives?
Yeah.
I don't think they're going to be really large scale clinical trials to test that. And so the adoption of them might take a while. And we've got to see how that plays out, kind of maybe smaller studies. And you can make interventions based on them, but maybe if you're going to ask people to intervene themselves in their lifestyle, I'm not sure how successful that's going to be.
Yeah.
So we'll see. They're definitely predictive, and we should make investment in that area. I mean, from a pharmaceutical perspective, developing drugs, yeah. I mean, stratifying people according to risk, looking to see whether treatment effects are different in different groups is really important. And I think us and other pharma companies are starting to do that. And then choosing people for trials. When you're trying to design a trial, large scale clinical trials are very hard and expensive. And careful selection of people using these tools could move the needle, I think.
And is the idea there to select people who are faster progressing or have a higher event rate of some sort, just to enable you to run a smaller trial? Is that right?
Yeah, potentially. Yeah. I think depends on the particular disease and drug, but yeah, I think that's the broad idea.
Yeah. In that paradigm, what does do you run a risk that what you learn from the smaller trial with the high polygenic burden patients doesn't translate to a broader population when you run the big phase 3? Or is that an open question? Can what we know about the way polygenic risk scores work tell us something about that, or is that a TBD?
I mean, I don't work on this directly, but I imagine that you kind of want to understand the effects across the PRS spectrum. You don't just want to choose all the people in the highest decile. You want to kind of understand how that changes as you increase your polygenic risk score.
Yeah, but I'm thinking out loud here. Maybe you run a trial that has people in the top decile and people in other deciles, and you stop early and you understand the trends in the broader population, right? But you then use that to make a decision on the phase 3 rather than. I'm definitely out of my lane here exactly on how this would be applied, but I'm curious about it because I hear a lot of people talking about using them in clinical trials, and there's a couple examples that I'm aware of, but it seems to be one of those things that is probably going to be another couple of years before we really start to see it pick up steam.
Yeah, I think I'd agree with that. I mean, I'd be quite surprised if there are effects that only occur in the highest decile. I think there's probably going to be a gradient.
Yeah. How about looking forward? We've got, as we talked about, millions of genomes, millions of exomes. What's the missing piece today in your mind? And maybe there's another influential paper that you've been part of that looked at comparing whole genome sequencing to exomes plus genotype arrays. I would love if you could just comment on, I have this simplistic worldview right now that maybe whole genome sequencing is for rare disease diagnostics and exomes and genotype arrays are for everything else. But I love your perspective on how we invest going forward in these different technologies.
Yeah, I mean, I guess I'm a bit biased. At the RGC, we kind of really believe, and we published this paper, like you say, that exomes plus augmented with imputation gets you almost the same data set as you would if you had whole genome sequence data, and of course is a lot cheaper. In our hands, when we look, comparing that approach to whole genomes, the yield is very, very similar in terms of associations. And of course, if you invest the same amount of money, you can do many more samples, and sample size rules, right? You can do 3 to 4 times the number of samples. You can't beat that. And so imputation gets you, in both exomes and whole genomes gets you the coding genome. I mean, there are small differences, but they're pretty small. And so you're kind of fiddling around the edges really there.
Yeah.
And then what's in the non-coding space? Well, imputation gets you the larger the reference panel. And then, of course, there aren't large reference panels in all populations, but we think you can still get pretty low allele frequency. And then what's left, what whole genomes gets you over that exome plus imputation approach is the really rare non-coding. So that's a big investment of money. And then, of course, these things can be very hard to interpret. They're not, the beauty of exome sequencing is the interpretability of the findings. And I've seen that play out with Regeneron. Our leadership really clearly, it's really clear what these association studies mean when you effectively knock out this gene in someone. But when you've got a non-coding variant, it's not clear what's going on so much. So, and I guess, I don't know, I haven't seen this flood of papers. The UK Biobank whole genome data has been out for a while. I haven't seen this big flood of papers with all these rare non-coding findings. I mean, maybe people are sitting on them, and maybe the companies who invested in this have them. It'll take a bit longer for us to understand that. So, but it's a valuable data set, and you can do lots of other things with it. But like you say, for rare diseases, but for our drug target discovery purposes, we're pretty happy with our approach. I mean, it's not that we are doing some whole genome sequencing, but we're not abandoning the exome sequencing. I'd be really surprised if we did that on my decision, of course. But I think it was a really inspired decision for general leadership to invest in this direction. And we published some of our findings. It's clearly been the GPR75 finding for BMI obesity, led by Luca Lotta and all my other colleagues. That's a really interesting protective association for BMI, on average 2 unit BMI decrease when you can, for heads in this gene, 5 kilos of weight loss, 50% odds ratio for obesity. That's something we published, and there's a drug program now, Regeneron. There are other examples as well. It's incredibly exciting to watch these move forward. I think that's one of the things I found really positive about moving from academia to industry, this ability to work with this incredibly talented group of scientists and with the technology and resources to move these findings forward and see that play out. And I've learned a lot. It's definitely not making the association finding isn't the end of the story. There's a huge.
No, it's the beginning.
It's the beginning, but it's been really nice to watch some of these stories move forward and learn about that.
You have spent and do spend a lot of your time thinking about machine learning and AI. I'd love to get your perspective on, won't it reshape what we do today? It's obviously taking the world by storm, but I would maybe betraying my ignorance here, but many of the underlying models that we use in genetics are surprisingly simple just because of the structure of the genome. So I'd love to have your perspective on that.
Sure. Well, I mean, like you say, it's really exciting. I mean, these models, deep learning models, AI models, a lot of them, I think the development came out of, image analysis problems, spot things and classify things in images, convolutional neural network models, basically allowed us to solve highly nonlinear problems, right? And these models were around, but we worked out how to fit them quickly and scale them up so they're very large. It's really impressive. But, not everything is highly nonlinear. Like you said, it seems like in things like polygenic risk scores, proteomics, risk models, things are surprisingly linear. There's not much benefit to going beyond that. I don't work on that, do it so much directly, but that's my sense of reading the literature. I mean, in general, I guess the way to think about this is there are lots of statistical methods out there. You can call them statistical methods or machine learning or AI, kind of the same. These new models and methods are some great new tools to have in your toolbox. And so I guess the way I like to think about it is start with the scientific problem you're trying to solve. And then do you have the data that I might need to answer that? And then it's a combination of the data and the method. You don't really know that up front. Before PRSs took off, you might think that this is highly nonlinear system involving
Well, a
all
lot of
these.
people did, right?
Yeah. I remember working with a PhD student back in, gosh, what was this? 2010 on trying to predict type 2 diabetes. Of course, much smaller sample sizes from using the neural network model. Of course, then we had, because of the smaller sample size, I don't think we had the power to detect these nonlinear effects so much. It hasn't really changed. So, like I said, what's the research question? And then it's thinking about the data and the model, and you don't jump to the most sophisticated model straight away. You might be able to get away with a much simpler and maybe more interpretable model. But having said that, these models we are using them in some instances. As part of my group that work on image analysis, we take the UK Biobank imaging data, all of the modalities, and they're extracting phenotypes from all of those. And we use my team use deep learning models to do some of that, not all of the time, but it's transformed that space. Yeah, what you can do, what you can extract from images, and so that's been very useful both for discovery and when we make a finding from a more traditional phenotype. What does the imaging show? And so even though the sample size in biobank is a lot smaller, the phenotypes are kind of maybe better. Yeah, if I want to measure, if I make a discovery using BMI, which of course is cheap and easy to measure in large numbers, but then I look at how fat is distributed in the body from MRI, or DEXA, then I can get maybe more specific signal. And then we've got a preprint recently on trying to use the proteomics data in biobank to enhance some of the large language model annotation approaches that exist. So taking those models like ESM1V and kind of tweaking them effectively in a transfer learning approach, using the proteomic data to improve the annotation. And we see that yielding more associations when we do burden tests. So there's definitely excitement and interest and applications. It's just, like I say, I don't think it's the solution to everything. But, these new tools exist. You have to learn how to use them. And I think my colleagues really like learning about that. And there are lots of talented people out there who are really good at applying them. So it's an exciting time.
Yeah.
So I'm positive, but just not totally going home. I think
not over your skis
is good.
Yeah, I suspect it's going to be very domain dependent, right? When I think about some of the challenges that we do have today is interpreting a variant of one, right? You've only got one person or one instance of it, and there's been some interesting work from the EVO2 model, it's a DNA sequence based model, and it hasn't been fed any information about pathogenicity of variants, but they applied it to test BRCA1 variants and was able to predict extraordinarily well. Actually, I was kind of surprised, and I think the protein models that you talk about probably could be very good at this kind of problem, right? What do we do when we don't have millions of iterations of people and we've only seen it once, or maybe the generative applications, right? A synthetic biology type approach. We need to generate something from scratch, but I struggle to see it a little bit in the complex trait world just because of how linear it is outside of phenotyping, of course, like you mentioned.
Yes. It probably, I don't know, I guess I would think about it probably is quite non-linear. It's just our phenotypes are kind of noisy sometimes, and our environment is very heterogeneous. The literature in the kind of mouse model organism world there's all of these interactions happening. Yeah. But the environment is controlled. And then, if you go to a setting where the environment is heterogeneous, I think that kind of might destroy your ability to find those linearities.
Yeah.
And that's not something we can solve easily. You can't do mass experiments in humans also.
Right.
Phenotype data is always a challenge, right? We can the phenotype of a human through their life is, much, much more high dimensional than in a genome.
Yeah.
And it's really hard to measure that accurately. I think we could be great to have more longitudinal data on people many years, but that comes with challenges as well. But for drug discovery, being able to develop biomarkers you can use in trials, I think you kind of need longitudinal data in large numbers of people to do that. And it's going to take time and investment. Yeah, I mean, I'm pretty excited about proteomics. And of course, you'll be aware that now on the biobank is going to be have the assay run, and of course, the extra 100,000 people who would take part in the imaging experiment, an extra collection, a second time point. So the potential for that to drive discovery and also augment what you know about existing things you're interested in, I think will be huge.
Yeah.
So we'll see. Maybe when we've got 500, 600,000 people, we'll be able to pick up some non-linearities. I don't know, but it's not looking.
They're not jumping out yet. So Jonathan, this has been amazing. If people want to follow your work more, obviously they can look you up on Google Scholar, but is there anything else that you're looking for right now? Are you hiring? Are you looking for more collaborations? What would be most helpful to you from the audience?
Yeah, from the RGC perspective, we're always very happy to talk to people who'd like to collaborate. I mean, please reach out to me or any of my colleagues, and we'll set up conversation, read our papers, and I hope they give a good representation of how we operate. Yeah, there are various. I think the RGC has various open positions at the moment. I'm about to, I think maybe on Monday, to put out a new advert for a job for someone to join my team.
Amazing.
But this is kind of we've grown very quickly, I guess, over the last 7 years I've been at Regeneron. I think that's kind of plateaued a bit. We have this amazing team operating really well, but we definitely are growing to some extent. We work on a kind of yearly cycle. At the end of each year, we have to ask for positions and make a case for those, and then, but it's some people leave and positions open up. So yeah, please free to chat. I guess I'm not super active on social media. We try to.
For the best these days.
Yeah, maybe. Yeah. Well, absolutely. Yeah. I mean, we use social media to advertise some of the work that we do. So watch the Regeneron on the RGC feeds. Yeah, but thank you again for this. It's been really great to chat.
Wonderful. Yeah, thank you for taking the time and for all the foundational work you've done over the years.
Thank you.
All right. Thanks, everyone, as always, for listening, and we'll see you next time. That wraps up this week's episode of the Genetics Podcast. I'd like to give a huge thank you to our guests for sharing their valuable insights and experience. And thank you as well to our listeners, as always, for tuning in. If you enjoyed today's talk, the number one thing I would really appreciate from you is if you could share it with a friend or colleague who you think would enjoy it as well. We would also really appreciate if you could subscribe to our show and give us a quick rate and review on Apple Podcasts, Spotify, or your favorite podcast platform. Both of these things help us become more visible when people search for genetics and precision medicine podcasts. And we're always eager to hear from you. Please reach out to us with any questions or feedback on social media. You can find Cynogenetics on Twitter, LinkedIn, or visit cynogenetics.com. Finally, a big thank you to the team behind the scenes who make all this possible, in particular Amy Cousins and Sonia Shah, who produced the show, and James Pierce from Selected Frequencies, who handles the audio engineering. I'm Patrick Short, your host, and it's been a pleasure. Thank you, and we'll see you next time on the Genetics Podcast.